Good morning, everybody. Welcome to today's webinar. My name is Jess Patterson. I'm a Senior Director BD here at CKDM. On behalf of CKDM, I'd like to welcome everybody. Today's webinar is a kind of point-counterpoint style, discussing machine learning versus physics-based approaches for computer-assisted drug design. As you know, or as you may not know, or if you do not know, we hosted a series of 3 webinars in the last kind of year and a half, where we explored the implications of artificial intelligence and machine learning in the drug development space. We started off with one looking at its implications in target discovery. We then did one on drug discovery and another one on kind of implications into drug development or clinical development. And so we thought we had some really great discussions last year. If you're interested in any of those or did not participate in any of those, the videos are available on the CKDM website at ckdm.org. But in the meantime, what we'd like to do going forward is to kind of maybe do a little bit of a deeper dive on specific subjects within some of those larger areas. And so we're really happy today to be welcoming Philip Tagari back as the moderator for these sessions. Where we're really going to look more at, as we said, this computer-assisted drug design and the approaches and pros and cons and balances of machine learning and artificial intelligence versus physics-based approaches. So again, welcome on behalf of CKDM, and I'll pass it over to Philip Tagari. Philip?
Okay, bonjour tout le monde, and good morning, everybody. Thanks to those of you on the West Coast who got up early, and thanks to our colleagues who are listening in from Europe in the evening. This is an incredibly timely session. 2 weeks ago, I was very fortunate to, Incitro was very fortunate to receive a visit from Eric Horvitz, the chief scientist of Microsoft and the champion of the collaboration between Microsoft and OpenAI. And he spoke extensively about his recent publication called Sparks of General Intelligence. And as part of our discussion, we spoke quite a lot about the ability of large language models to start to think chemically. And so it's an incredibly timely opportunity then to meet with 4 of the most sophisticated and accomplished practitioners in the field of drug design and computation, where we can explore now the impact of AI and machine learning on the business of small molecule drug discovery. So without further ado, I'll ask each of the panelists in turn to introduce themselves with a short background, and then we'll have a series of short presentations and then get into discussion. And so I'll start then to ask Jennifer to introduce herself.
Okay. Hi, good morning, everyone. I'm Jennifer Bui. I'm currently a senior scientist at Microsmith Laboratories at UBC. I am a truly multi-scale modeler. I started out with quantum mechanics. Now I'm doing a lot of bioengineering. So I got my BS in physical chemistry, doing quantum mechanics, and moved out to San Diego in the lab of Professor Andy McCammon. After that, I got the NSF postdoctoral career fellowship to go to England in the lab of the late Sir Christopher Dobson's to study protein folding. And I moved back to the US to join David Baker Lab, and I was appointed as research assistant professor at the Quantum Institute at UCSD, and I was also appointed as an adjunct professor at the Pharmaceutical Sciences at UBC. I know I called Alan Bart on one of my response for the physical-based model. It is a cheap shot, actually, to get you guys out of bed to listen to the webinar. The truth is that the computational power available today is far surpasses the computational power that back in 1980s when Alan Barr gave a seminar about physical-based modeling. So I'm excited to be here to talk about new recent development in the machine learning algorithms and force fields. Thank you.
Excellent. Thank you, Jennifer. And Anthony.
Good morning. Despite your introduction as being sophisticated and practiced, I don't feel like I've quite hit that bar yet. But my background is in biochemistry and bioinformatics. I've been fortunate to have been able to practice in bioinformatics for probably close to 20 years now. I started with undergrads at Waterloo and finished my grad school, both a master's and PhD at UBC. But since then, and even before finishing my PhD, or before I started my PhD even, I was fortunate to be one of the founders at Zymeworks. So my background in this space goes back at least that far. That said, really the premise of the company that I'm at, H2O Biosciences, where I'm the CEO, is that we think that there's a lot of room for improvement in the physical models, and those force fields are, as Jennifer said, they came out in the 1980s. There's a lot of room for us to improve, and my focus, at least since starting Zymeworks, has always been on the idea that we should be improving those force fields, and we shouldn't be ignoring the physics. So that'll probably be the basis of most of what my perspective for this discussion.
Excellent. And then our next panelist, Tom.
Thanks very much, Philip. I'm Tom Miller. I'm one of the co-founders and the CEO of Entos AI. I come from a background of physical chemistry, did my undergraduate at Texas A&M before going to Oxford on a Marshall Scholarship to do reactive scattering in the gas phase, and then began a steady progression towards statistical mechanics at Berkeley, and then became an assistant professor at Caltech in 2008, and then went through the ranks of full professorship, and spent 14 years there as a faculty member, working at the interface of new physical modeling and the application of those to various chemical and biological applications. A couple of years ago, we had the opportunity to see the translational opportunity of some of the AI and physics-based tools that were being developed in my group, and we created Entos, a company that's committed to using physics-informed AI in combination with high-throughput experimentation to accelerate small molecule therapeutic design and discovery. It's been an incredibly exciting journey, and I'm very happy to share what we're up to.
And finally, Alan.
Hi, so my name is Alan Chang. I lead a group. I'm a senior director at Merck, leading a group that works on lead optimization of both small molecules and biologics, as well as about 5 years ago, set up a subgroup that works on deep learning algorithms for lead optimization. I'm based in the Bay Area, so kind of took advantage of the excitement and enthusiasm around deep learning then. Before Merck, I was at Amgen with Philip. It was a great time. I was there for 11 years. And then I was at Pfizer before that. And then before that, I was in grad school at UCSF, and then in biophysics and undergrad degrees in electrical engineering and computer science and molecular biology at Berkeley. Looking forward to the discussion today.
Excellent. So again, a big thank you to all of the panelists for taking their time today. Thanks for everybody joining. And then just a reminder of the format, we're going to have 4 short presentations. If you have questions during those presentations, please type them into the chat. And then hopefully we can get to them during the panel session after the pre-submitted questions. And so our first talk today is from Jennifer.
All right. Can you move to the next slide? In interest of time. Again, I already introduced myself, so next slide, please. It's very blurry. Can you see my the PowerPoint? The presentation is very blurry on my end.
Yes.
Are you guys seeing it okay?
No. Can you share from your desktop, please?
Yes. Can you allow me to share? I can't. Can I? Okay.
Perfect. Thank you.
Perfect. Awesome. So our research has been focusing on intrinsically disordered protein, short IDP. IDP lacks a unique three-dimensional structures. The order of structuring can see on this slide here. Approximately 50 to 70% of our proteins in human genome associated cancer, diabetes, and neurodegenerative diseases has at least minimum 30 residue that are intrinsically disordered. Therefore, IDPs form an important class of therapeutic agents. Our pipeline seems quite diverse. As a co-founder of a startup that leverages AI and physical modeling, we aim to develop methods that advance the field of drug discovery. So I put a website here. This website, you please go and visit and explore the tools that we have developed from identify the disorder protein binding region to predict the mutation that could be deleterious. So most of us by now have used AlphaFold2 to obtain protein structure of your interest. But there are other AI-based programs like Rosetta 4, for instance. So these are my favorite fields. So how well AI-based structure prediction of proteins with the long intrinsically disordered regions, that is, I say, about more than 30 amino acids. Well, you can see in the sample that I show here that the GDP43 that is known associated with ALS, androgen receptors associated with prostate cancer, and the S1 transcription factor, which I talk a little bit more about. These structures are predicted by AlphaFold2 because I figure it's easier to pick a fight with John Jumper than David Becker, or the other way around. Anyhow, you can see this as the disordered regions are predicted at very low confidence, and AIs for the disordered regions, doesn't mean that they don't have any structure, but that they have an ensemble conformation. So the physical base methods such as molecular dynamic simulants can help to sample the ensemble conformations of the protein disorder region. So the fact it can help is, you can see is here is the energy landscape of the physical base model for the other protein are quite different from the disorder protein. For the other protein, there is a kind of funnel-like energy landscape, whereas the disorder protein, you see that there are multiple states and they are coupled together by very low energy barriers. In fact, a paper by Suzanne Nuchuk recently showed that these energy barriers is in the thermal fluctuation is about 2 kg max. So using long MD simulations, for instance, you can obtain the ensemble conformations. That is not to say that the physical base force fields such as amber, jam, using the various water molecules as shown here, could accurately capture the ensemble conformations. In fact, they tend to sample a more compact and more helical structure. But there are also recent paper by Ray Liu lab that one of my favorites force fields to use for IDPs, the FF14 IDP, and you can, the picture is just shown here that is kind of accurately or not, in somehow, in agreement with experimental data. So the physical model-based model that on the transcript factor is that most of the transcript factors belong to the S family domain. It's have a two-part. One is the DNA binding domain, and the other side is the disorder part. This is the disorder part often come and bind to interact with the DNA binding parts. Therefore, the term is also inhibitions. We recently come up with a protocol that combined Rosetta and MD simulations to obtain the structural ensemble of disorder region of this transcription factor, the S1 that I shown here. Take my word for it, but I do not have time to go through the protocol, but regardless, the confirmation that we started that at the center of the cluster, that we could find that the trajectory, that we started, from MD trajectories, that it would visit the stage with auto-inhibit the stage that is when the residue, one of the residue from the disorder region part could come and interact with the DNA binding part. This video just showing that how it moved one of the stage that is in the quite close to the final stages where the auto inhibition occurred like this. So it's, we move it back a little bit. So it will move or would visit it or sample the conformation where it's really much look like the auto-inhibitor stage. Along the line that we could see here is the final products that we compared in the trajectory of the inhibited stage. That as you see that these integrated disorder part come together to form the allosteric pocket denoted here in green, and that the DNA binding site. The moving quite long, but anyhow, take my word for it. I'll go move there. Otherwise, I wouldn't put it there. Anyway, so the discover of the allosteric pockets is could be good to can be explored to screen for small molecules that mimic these interactions. So this is the protocol that we use for physical-based screenings. I don't have time to walk through it, but it's really much like the relaxed complex method developed in Andy McCammon lab. That by the end we screen the NIH small molecule depositories, about 500 compounds. And there are two classes of binders. One is mimic the DNA binding, and the other one is occupy the allosteric pocket. The protocols, we compare these protocol tests with using NMR, and these are the the HSQC NMR titration, the chemical perturbations, which shown that when the outer inhibited state where you titrate in is the phosphorylated disorder part to the DNA binding domain, they do bind. So the compound number 9, which is quite an M-compass then between the allosteric binding site and the DNA binding site. So it's quite a long molecule, and it's also mimicking. It's really much the same. Like that of the disorder recent part NMR titrations. And we also compared, and how do we know that the chemical, the physically binding interaction that occurred that are similar. We use the method called the community network analysis, which is kind of neural network clustering. One can see is that we denote as a group of atoms that acting in a constant motion as a node, and the interaction those group of residues or atoms of the protein as the edge between them. And you can see that the titration of disorder part to the DNA binding domain and the compound number 9 to DNA domain, that's exhibit similar actions, kind of network interactions. This work is done by Cecilia. How much time do I have? Anyway, so in the so what I'm very was quite excited. We all get very excited about the new diffusive generative models, right? In protein structure prediction, Rosetta diffusive just published recently, and this is the quite cool program. You should totally check it out if you want to do protein designs, protein folding. On the other side is the deep dark, which is a program use a diffusive generative model for the small molecule and protein interactions. And Gabriel has helping me try to also try to screen in this kind of allosteric pockets for small compound, and deep dark, but it's really fast, and it's captured quite a number of novel active site. And see the result. Deep dark here is not only binding to the DNA, the allosteric pocket, but also another a kind of novel alternative pocket over there. So these are just the comparisons between the NMR model, the Rosetta dark and deep dark. So but like I said, it's a great method for screening a small molecule and try figure out the novel binding site. Totally recommend deep dark. As closing thought, I'm quite excited about a new machine learning for fields that I'm fortunately collaborate with our college here at UBC and friends from Cambridge. Just to you the what it can do, and you can see here's an example of the potential surface of the G-bench log C's pyrimine to amine, denotes A3PA, and as Maze is really with the multi atom center clustering come quite close to the first principle calculation from the DFT. So this is really quite exciting that for the first time that the machine learning for fields capture almost, I'm sorry. come very close to the first principles test. So we hope that we could combine maze and deep dark for screening of small molecules against our IDP targets. With that, I stop here and thank everyone for your attention.
Philip, you're on mute.
And with that, I'll thank you, Jennifer, and I'll pass the podium over to Anthony. And if we could have the main presentation back.
Right. Thank you very much, Philip. I have a little bit of a different tack than Jennifer. I didn't bring much data with me, but I'd like to introduce you a bit to the company H2O Biosciences. I think what we're working on is pretty relevant. I've already sort of covered most of this in the introduction, except to point out that my daughter in the picture would like to remind everybody that my dad jokes are not funny, so you probably shouldn't be laughing at anything I say, but I'll leave it at that. The company that I've started along with my co-founder Alex Shinkarenko is H2O Biosciences, and sorry, is that covering the slide? Maybe a little bit. And the idea behind the company is basically to rebuild all the physics from the ground up, because I think what we saw in Jennifer's presentation is that there's also an opportunity for people, or there's the constant requirement for people using molecular dynamics and physics-based programs to tune their force field or look at different force fields that solve different problems. And as exactly as she said, sometimes we find the existing ones force helical structures or sheet structures where they shouldn't. And there's a lot of discrepancies between where reality lies and what the physics-based tools have been showing to us for a long time. So in any case, the company we started, we're now 11 people, and our focus has been on designing. Oh, the presentation seems to be going pretty wonky, just for the technical team at the back. Nonetheless, I'll carry on. So basic idea is that we want to be able to work on cheaper, faster, and better drug design. Normally in the world of software development, you pick two of those, cheaper, faster, or better. We actually think there's an opportunity to go after all three at the same time. The company is looking for strategic R&D collaborations. That's our goal for the end of the year. And we're just now getting to the point where we have a tool we believe will be commercially usable. Yeah, I'm not sure how much you can see on this slide. Probably not a whole lot. Nonetheless, I can walk you through what's here. At the very top, we have a traditional force field. So pretty much all of the existing tools out there use the same formula. And regardless of whether you can read it or not, it basically has 5 terms, and basically all 5 terms are wrong. There's one that describes how bonds form, or rather not form, bonds are held. There's one for angles, so in the midst of a single atom, how its angles between the connecting bonds will work. Dihedrals, Leonard-Jones potentials are basically how much atoms want to repulse each other, and charges. In the equation that exists, you see a lot of things that are pretty much set in stone from the day that these equations were written in the 1970s. Bonds don't break, charges don't change, and the atoms are sort of fixed in their properties. And all of those are effectively not describing reality. So our ability to extrapolate from this basic equation to complicated systems like intrinsically disordered proteins is somewhat compromised. We can get great answers out of it, but it's not always going to give us the real answers or the whole answers. And so fundamentally, we have to go back and revisit that. And that's what H2O has done. We've studied protonation and deprotonation. So the ability to break and reform bonds is important, particularly when you're talking about solvent. Water is always constantly moving about and doing interesting things. And if you're talking about pH, that literally is passing protons between different components of the system. So things like that are really important for us. We also talked about charge fluctuations for the last 40 years. People, sorry, Jesse?
Do you want to try and share from your end if it's set up anyway? Just because it is very it's really not clear on the screen, and there's not sure what there's a technical issue on the back end of something. But
yeah, it's pretty bad. I didn't keep the slides open, so you'd have to give me about a minute to go track them back down again.
Oh, I mean,
sorry.
Okay, go ahead.
Yeah, we may as well just carry on. The pictures aren't particularly important anyways. I can walk through them as best I can in any case. So the thing that, oh, there they go. Now they're a little bit better.
Yeah.
So fundamentally, I think what really we need to do is build a better model. And a better model should let us do protonation and deprotonation. It should let the terms interact. In a normal chemistry experiment, if you squish a molecule together, the way that it twists and turns and interacts should change from if it's relaxed and if the bonds are longer. And those things don't happen in the existing models. They're really fixed in from the beginning. And charge fluctuations have been worked on for the last 40 years. Nobody's really been able to do a fantastic job of integrating them. And that's something that we focused really heavily on. So we believe our model is capable of doing these things. And what we should be able to get out of this is better emergent properties. And the inability of traditional models to get emergent properties out of these systems has actually been a great reason why I think AI has picked up in this space, because we simply cannot get the full answer out. And there was a hope that AI might be able to see the answers where the physics couldn't. Fundamentally, I believe that if you get the physics right, then you should be able to get those out. Whether you're using AI, whether you're using physics-based models, at the end of the day, you do need to see what the system's really doing, and all the pieces that are subtle should translate into real chemistry. So for us, we see it as a set of building blocks or a set of layers. Fundamentally, you have to get the physics right. If you get the physics right, that's going to take you to the next layer of getting the chemistry right. If you get the chemistry right, you should be able to see great biology coming out of that. So we've built what's considered a polarized and reactive force field. It's definitely unique in the space. No one else has done the same type of work that we have. So we have something that's hopefully going to change the way that we see the chemistry. If you can do the chemistry right, you should be able to do better surface chemistry. You should be able to pick up binding site pHs. You should be able to understand charge sites. And hopefully at the end of this, you should be able to see allosteric changes. You should be able to see, if you've got all these pieces right, the protein should react the way it should react in a test tube, and you should be able to see that. And that would allow you to do a whole lot of different biology, from cyclic peptides. I saw a question about cryptic binding sites. All these sort of things come out of having the right chemistry. So I would propose that whether we get there with AI or whether we get there with physics-based, both of them will have a long path in front of them, and only by walking that path are we going to get there. Oops. Okay, so I'm just going to talk briefly. I didn't bring any data slides, but I would like to tell you at least a little bit about the project we're working on. If you look at, slides keep disappearing. If you look at our project versus something like Amber, which is a great competitor and frequently used in this space, Amber has to over-parameterize everything. There's 22 different types of carbons that they consider. So a carbon that interacts in a planar environment is different than a carbon that interacts in an amino acid. That's different than it interacts in a long chain. That's not how the chemistry really should work. We don't believe that that's the way it should be done. In our platform, we talk about sp2 carbons and sp3 carbons. And from there, all the different properties of those carbons come out. And we're still able to do the same things that every other platform can do. We just use the chemistry to inform us and get that right. So our model appears to be much more consistent with the basic chemistry. We can talk about that in different ways. You have different metrics for showing it. I didn't bring any slides to talk about those metrics, but I would say that at the moment, our platform is already at par with some of the other ones out there. We can talk about MMFF or Amber, any of those. And we're basically showing our benchmarks using the generalized Amber force field test, or similar tests are showing that we're doing this the right way and we're getting the same answers that they are. And one of the big issues that I think we see, particularly around stuff like intrinsically disordered protein, is really understanding torsional profiles and getting all those interactions right. So we can show that we're starting to get those with much less artifacts. So I'll leave it at that. If anyone's really interested, you can follow up with me afterwards. I'd be happy to share one of our decks with you that has more information in it. Just wanted to touch a little bit more on the subject for today. I really see artificial intelligence versus physics-based as doing two different things. We can talk about artificial intelligence, which is really fantastic at interpolation. You give it a really big data set, it should be able to understand the data set, what fits into it, what doesn't fit into it, what belongs, and the generative AI should be able to predict things that belong in that set. However, it might be more difficult to get it to generate things that don't belong in that set. If you haven't trained it for a specific function, it's going to be hard to get that information out of it. If you wanted to work on cryptic binding sites, that I know that's something that we're probably going to touch on later, that'd be really hard to do with AI because you haven't got a data training set for it. On the other hand, physics-based is a little bit different. We have a model, we can test new theories, we can ask questions that otherwise we don't have a training set for. You can decompose it, understand how the pieces work, and then you can ask where the model breaks down. And I think that's really important. One of the things that I most worry about with AI is the ability for it to what's called hallucinate. And I'm sure there's probably people here who know much more about this than I do. But when it starts to give you answers that it doesn't know are real, how do you differentiate? A physics-based model will always be able to let you understand where those boundaries lie. And I think that's really key in when you're starting to walk out into the world of stuff we haven't really worked on in the past. I also don't think that they're competitive. I see them as complementary. Generative AI is going to be the best tool for us for upstream discovering new molecules. But at the far end of this pipeline, you still need to do optimization. To take a hit to a lead, AI is going to be much harder to get you there. You have to break things down and be able to generate insight. And AI doesn't always do that. I think that's where we'll see the competition. So that's where we've been focused. Just want to quickly end by saying thank you to my team. All the work that we do is not powered by me, it's powered by my team. And in the last 2 years, they've managed to accomplish a lot of things that have been open questions for the last 40. So a quick shout out to them. That's it. Pass it along.
Excellent. Thank you so much, Anthony. And I'll then pass over to Tom from Entos.
Thank you very much, Philip. And I'll request access to share my screen so that we can have fewer such iterations. Can I please have access to share my screen? Great. Thanks. Okay, wonderful. So that should look okay, I hope. Yeah, it's very exciting to be part of this panel. It's wonderful to be part of the discussion and to consider the advantages and strengths of machine learning versus physics-based approaches for CAD. At Entos, what we're really focused on is the delivery of small molecule therapeutics to clinic in a way that provides the ability to deliver molecules for unmet need, highly differentiated molecules, and to do that at a pace that gets them to the patients that need them very quickly. And one of the generic strategies, the broad sweeping strategies that we employ is to try to take things that would normally be tackled in sequence, things that would normally be decomposed into stages like hit ID, lead optimization, and to, as much as possible, not deal with those in sequence, but to tackle those through parallel consideration as early as possible. Right out of the gate, consider factors like PK, consider factors like synthetic accessibility for CMC and toxicity. And the goal of that is to therefore become less committed to chemical matter that we later find to be problematic, and to find those regions of chemical space that are highly differentiated, and to do that as quickly as possible. And we use both AI and physics-based modeling, of course, for this pursuit. So kind of thinking to, just the general, framing of the discussion today, it's of course a very natural way to compose a panel, around the question of machine learning versus physics-based modeling. But as several of the speakers have already kind of pointed to, I think it's much more a question of how machine learning and physics-based modeling must work hand in hand in order to kind of pursue these goals and to address these challenges. And there's kind of an obvious way in which that works. And then there's maybe a slightly less obvious, but also interesting way in which those two things can work together. That's what I want to point out here. The way in which I think we all naturally think of physics-based modeling and machine learning work together is just by saying that some are good at some tasks and some are good at other tasks. Anthony was pointing out the fact that, for generative AI, you can do that for some early tasks, but physics-based modeling will help you with other things, perhaps at the optimization stage. And kind of a division of labor between things that are, data rich in terms of what you can tackle and things that are data poor, where physics should be striving to supplement that. But we also think about that in a second way. We think about that in terms of the fact that machine learning models themselves, the actual data-driven predictive models themselves, are made better when we incorporate physics into the way those models are constructed. That makes them intrinsically more data efficient and more powerful in their predictive capability. And that's something that we've really tried to embrace for quite a long time, both in my academic career at Caltech and also at Entos as well. And one really easy way to see how physics-based modeling, better physics, leads to better AI is kind of through this very simple illustration on the left-hand side. If you're trying to do a machine learning model that will recognize triangles, you could basically show it triangles of all different orientations. And if I don't know the fundamental symmetries of rotation of a triangle, I would have to see lots of those images. I would have to have a very data hungry algorithm to recognize, when I see another triangle by somehow interpolating between these different things. On the other hand, if I build a machine learning model that naturally incorporates that rotational symmetry of triangles, well, I only need to see one such example before I can very successfully predict and recognize subsequent triangles. And that goes not just for simple geometry like that. Molecules are actually a generalization of this idea. Molecular orbital structure is indeed something that incorporates a huge amount of symmetry. If you build machine learning models that see a molecule as a carbon linked to a carbon linked to an oxygen, where each of those oxygens are different types and each of those carbons are different types, you have a combinatorially large number of ways of linking those together. And much like this case with the triangles, you have something that is intrinsically very data hungry. On the other hand, if you can abstract the atom types, if you can represent the molecule in terms of the underlying molecular orbital structure, even at the architecture level of the machine learning model, well, then you naturally incorporate hundreds of additional symmetries that would otherwise need to be learned from the data. But you should know those up front. That should be baked in there. And by incorporating those things, you can actually build much more data efficient models. We do that in various ways. We've written quite a few papers on the matter, and you can see those. I think two that are particularly interesting to this group is OrbNet. This is a paper that you can read about in the most recent report and benchmarking of it in PNAS last year, where what we do is we take the actual electronic structure, the actual machine learning, the actual molecular orbital structure of the molecules, we represent that on an equivariant graph. And then what we can do is then use that as the framework for prediction of energies and forces, torsional degrees of freedom, bond making and breaking, things that were mentioned in the other talks. And to do that at quantum mechanical accuracy, but with a much lower cost in terms of the input requirements. A second tool that I think is also relevant is one that's currently on the archive, which is Entos NeuralPlexer. Entos NeuralPlexer basically addresses the same challenge. I'll do a few more data slides on it in a moment, that we might think of as in terms of AlphaFold or RosettaFold, where you put in a primary sequence and it predicts a three-dimensional structure, but it does so in the presence of a small molecule ligand, thereby allowing you to predict cryptic pockets, to predict allostery, to predict ligand protein binding structures, much like DiffDock does as well. And this is a model that is, again, equivariant, so incorporating those natural physics. It incorporates representations that are inspired by multiscale physics, spanning from atomic resolution to coarse-grained resolution. It uses physics-based algorithms, such as incorporation of force fields within the predictive process, as well as Langevin dynamics for the actual way in which the generative solutions are arrived at. So again, but much physics is incorporated in the way in which we do these machine learning models to get better predictive accuracy and data efficiency. And we build those tools. You see AlphaFold here, you see NeuralPlexer here. We build those tools in a way that allows us to really tackle the tasks of drug discovery from the earliest stage of hit identification, mechanism of action discovery, and to carry that through to accurate prediction of pose and ligand protein binding affinity. We use very similar tricks in a tool that we call Propane to get very good data efficiency for the prediction of dozens of properties that are essential for lead optimization and multi-parameter optimization, spanning from physical chemistry to ADME to PK properties, and even to tissue distribution and toxicity. And we do that all within a framework that we call Magnet, which is a generative AI tool for predicting molecules that would be optimal according to the predictions of these other methods, and doing that in a way that's cognizant of our platform and the synthetic accessibility of our chemistry capabilities in a high throughput manner. So just to kind of show how neural plexure works in an example that I'm sure is very familiar to many in this audience, and including our esteemed moderator, the KRAS G12C example is a classic cryptic pocket challenge, one for which the identification of the location of the pocket is difficult by the fact that in the protein's resting state, it would be occluded. AlphaFold, having trained only on structures that are blind with respect to that conformational rearrangement associated with a cryptic pocket, places the alpha helix in such a way that occludes the pocket and therefore misses the appearance of that pocket and provides a strong steric clash. So you see the difference between what AlphaFold predicts in blue versus what the crystal structure would be in gray. Neural plexure with the same training data nonetheless is able to understand that intrinsic conformational flexibility of the protein, is able to understand the fact that this pocket is therefore available, and in a really nice way, without any kind of trickery associated. We just put in the blind protein sequence and a graph of the small molecule, and it's able to identify this the existence of the cryptic pocket and beautifully overlay that with the ground truth of the crystal structure. So this is, I think, an exciting illustration of what tools of this kind are able to achieve. We didn't just cherry pick this one example. Also in this paper, which is on the archive and which is submitted, we do this for over 100 different examples of cryptic pockets and ligand-activated conformations. We do this in data sets that are temporally split. So we only train on data prior to 2019, and then we only predict on data post-2019 to make that realistic with respect to the way actual research occurs. And we compare that against Rosetta Fold, and we compare that against AlphaFold. And very convincingly and across over 80% of different cases, Entos neural plexure is a more accurate prediction of the cryptic pocket and the resulting structure. And that's exciting because it spans multiple protein classes, multiple mechanisms of action, and a wide variety of targets of interest. Both AlphaFold and neural plexure are better than Rosetta Fold on this task. So, what we do at Entos is we take these tools, these various ways to design the molecules, we place those into a high throughput experimentation framework that allows us to design, make test cycles all the way from new molecule design through to actual data testing and retraining of the model and new data generation on a 7 to 10 day cycle. And we do that to drive all of our programs as quickly as possible to test more hypotheses and to de-risk leads and to arrive at better development candidates. And just as a final example of how that really works in practice, this is one of the 4 programs in our pipeline. This program is going from program launch to IND submission in less than 24 months on the basis of the fast process of discovery. It's HER2, which is an oncogene for which it's critical to get high levels of selectivity and brain penetrance. We iterated over the course of an 8-month discovery process, only having to synthesize 120 molecules in singleton and doing 14 sweeps through the high throughput experimentation platform in that process to drive through to superior potency, selectivity, and general ADME and PK and tissue distribution properties to arrive at a molecule that is very exciting and appears to be a best in class with 10-fold better selectivity versus the dose-limiting toxicity in EGFR, as well as 10-fold better brain penetrance than tucatinib. And what's exciting about this general approach is that this is the best molecule and the one that we've committed to for our development candidate. But from 3 different novel molecular series that generated from this search, we had actually over 100 distinct molecules that had over 100-fold selectivity against EGFR, giving us a wealth of potential candidates to choose from. So this illustrates in one example, we have other projects related to allostery and to protein-protein interactions, and we're using that to drive forward an internal pipeline with focus on oncology at Entos. So thank you very much for your attention.
Excellent. Really interesting illustration of AI and physics-based methods. And our final presenter today, before we get into hopefully a good to and fro in the panel, is Alan Chang. And you're muted, Alan.
Okay, I guess that would help. And you can see my slides?
Full screen, please.
Yeah, great. So thank you for joining and thank you for having me here. I will, like Tom, talk about, and Anthony and Jennifer before me, talk about AI/ML plus physics-based approaches. I'll give an example of Padme and Annie together. For those of you that know Star Wars, you probably understand this, and then I'll explain it a little later. So in the drug design cycle, many of you are probably familiar with design. You design the molecule, then you make the molecule, and then you test the molecule. And based on that data, you design your next round. In lead optimization, we go through this many, many times. And so there's increasing desire to go through these cycles less times. And you can do this by doing a design cycle within the drug design cycle. So design, predict, analyze, and refine your hypotheses before committing to making the molecule. Because making the molecule can be quite expensive. It's also resource intensive. And so to do this design, we can use computational biophysics, for instance, structure-based design, lots of the methods that you saw earlier, as well as AI/ML deep learning. And so for deep learning, it's a combination of compute power, algorithms, and data. And certainly at Merck, we have a lot of high quality, very consistent data that we can leverage to do that. And the hope here is that instead of going through this whole grassy field, you can find your way through in a little bit more direct way. And so drug design is essentially a multi-parameter optimization. You're optimizing not just the potency and efficacy, functional activity, but you're also optimizing for selectivity, safety, as well as all these ADME type parameters, including how long the drug stays in your body, so the mean residence time, permeability, solubility, because you want to be able to dose effectively. And then for CNS targets, PGP efflux is key. And so for these ADME models, we early on worked on graph neural networks with a collaboration with Stanford. And the paper is cited below. A student, Evan, actually spent quite a bit of time with me at Merck. And so we looked at the Merck data sets, and there are about 20 something shown here. And what we saw was, so performance, better performance is up. This is Pearson R squared on the y-axis. And these are the different assays on the x-axis. Random forest, which is our standard go-to, which was and partially is our standard go-to at Merck, is shown here using APDP fingerprints that were created at Merck. And then we have graph neural networks, 3 different types. So the first and second ones were the ones we created with our collaboration with Stanford. And then the third one is ChemProp, which is from the MIT consortium that Merck is also part of. Let me see if I can get a laser pointer. Let's see. There we go. And you see that the performance generally improves for all these assays, some more than others, especially for plasma protein binding. This log D improvement turns out to be pretty meaningful, even though it's already well performing at with random forest. But there are, on the other end of the spectrum, a lot of the in vivo data points, in vivo assays where it's not modeled well. And then this FASO solubility, we actually had a paper about the inherent noise in the model, or in the experimental data, I mean. So you can see for plasma protein binding, this is rat plasma protein binding, going from a random forest approach to a graph network approach, definitely a clear improvement in the scatter plot. Where it's pretty exciting, it was quite exciting to me, was when we did this molecular weight extrapolation test. So the previous one was a temporal split. This is where we do a temporal split, but we only train on smaller compounds and only test on bigger compounds. And you can see that the graph neural networks retain a lot of their performance in this type of test, this molecular weight extrapolation test, whereas the random forest approach tends to lose a lot of its predictive performance. And so this was, exciting and something really different than what we've seen before with ML approaches, and shows the potential for these newer deep neural network approaches. And so what we set out to do is further improve our models. And so we looked at QM, quantum chemistry properties, things like energies, polarizability, et cetera. The difficult thing is that these QM calculations are not practical at scale because they take hours per compound. And so what we did is we leveraged both this ANI learn QM potential, as well as our in-house GCNNs to predict QM properties. And a lot of this was, the literature was around carbon, nitrogen, oxygen, fluorine type molecules. And in drug discovery, sulfur chlorines are pretty frequent. And so we set out to extend, for instance, this QM9 data set to include sulfurs and chlorines. And then in ANI, they were doing something in parallel to extend this in ANI text. And so what we can see is, once we train up this model compared with QM9 by itself when applied to drug-like molecules. So this is go from 9 atom type molecules to drug-like molecules, which tend to be around 20 or more. You do see that this QM9 extension data set and network does predict better. We'd love for it to predict a lot better, but it does predict better, especially for the atomization energies. And so we applied these approaches to try to improve a number of our ADME models. And this is clearance, various forms of clearance. And then this is PGP efflux. And so if you just zoom in on PGP efflux, you can see with the standard ChemProp. And then by adding in ANI energies or further adding in QM9 extension data, you do see significantly improved predictive value from that. And so this is a case where we combine PADME, which is predictive ADME, and ANI, which is the force field, the learned potential, QM potential that you saw. And for those that are not familiar, Padme is the queen from Naboo and senator from Naboo in Star Wars. And then Annie is the boy from Tatooine who becomes Darth Vader. And so they had a conflict early on, but here we're bringing them back together. So having having part of that multi-parameter optimization largely on the ADME portion, coming back to thinking about the whole optimization process. And so I've been fortunate to have to help put 7 molecules into clinic, and just thinking about all that is involved in that. Here's one example of one molecule, MCL1, which is a protein-protein interaction example. And I think it's useful to think about this and all the different pieces that are involved. And think about how ML or AI ML can help, and what pieces are strongly physics-based. And so in MCL1, we started with a high throughput screen hit. This is a random screen, and we found a compound of 3.4 micromolar. got a crystal structure of that, as well as did NMR studies. And through just, evolved medchem, so just adding groups and trying things out, for instance, cyclizing this group or modifying this acid, and then combined with crystal structure that we were able to get in structure-based design, where we noticed that there was a space in the pocket that wasn't quite filled, and we want to link that up, and that's how you get this cyclization here and the spiral cycle here. We got to a point where we improved the potency quite a bit, by 100-fold. And then looking at the structure and looking at NMR data that shows that these two pieces were close to each other, we're able to link up the molecule using a variety of linkers, including the one shown here, that further improved potency and some of the properties as well. And then looking at this molecule in the structure, and then doing a conformational analysis, so looking at enumerating the conformations that the compound could have, the low energy conformations, and then kind of plotting this out. So this is low energy, and this is the Boltzmann population. You can see that this molecule itself was not the highest populated conformation, and so what we want to do is modify this compound so that it would be one of the highest populated conformations. So basically it's pre-formed for the binding site, and that would allow it to not have to have a strain penalty going into the binding site. And so we were able to rigidify this linker, and at this point there was a clearance issue, which was a lot of things were done, but this methoxy modification really helped. And so this is AMG 176, which is, I think, still in clinic, and is, we hope it's an important medicine for cancer. And so just as my last little box here, summary of physics and AI ML, thinking about this example as well as the Annie plus PADME examples, two possible approaches is that physics are good for low data predictions, and then you can use AI ML for knowledge-based design where you have a lot of data. The other approach you could do is do active learning, right, where you iterate between exploration and kind of using AI ML for exploitation. And then the second summary point is that physics is really good at those small changes because you have this physical model that's pretty good at extrapolation. And some of these moves that we had to make within MCL1 were fine-tuning to get half a kcal of improvement in terms of binding free energy. And where AI/ML could play a role in my mind is that it's good for hypothesis weeding ahead of detailed calculations. So physics-based does take a lot of compute time, as Anthony mentioned, and AI/ML could accelerate that. So, and that's my last slide. Thank you.
Excellent. So thank you so much, all of our participants, and thank you to those of you who are sticking with us online. And we have a good 20 or so minutes for question and answer. And so thank everyone who wrote in questions. I'm going to start off with a really interesting question, which is a little bit of a follow-on from my introductory remark. When we spoke with Eric Horowitz from Microsoft, he said that GPT-5, which will probably appear in 2025, will understand chemistry. And so the question that was written in was, large language models are very powerful, but often quite superficial in their knowledge. That to say, confronted against a human domain expert, the answer will be easily dismissed as trivial or in fact wrong, and the idea of hallucinating incorrect assertions. And so, the question for the panel, and maybe I'll ask kind of Tom to kick off, is, given that, large language models, genuinely are capable of learning through embeddings patterns that humans aren't, what do you think the possibility that GPT-5 or GPT-6 will actually outstrip physics-based models and the so-called chemist's intuition? And you're on mute, Tom.
Yeah. Glad to be able to kick that one off. So thanks very much, Philip. But it's a great question. It's the question of our time. There's no doubt that generative AI is changing content creation on all fronts, and chemistry will be no different than any other front. So it's coming, and it will be part of the landscape of what we see moving forward. I think that there's always going to be a process of there is something interesting between the way these models are used for maybe generating language versus generating chemistry. In some ways, all the words have been said before. All the sentences, in many ways, have been said before. So what they're doing is they're kind of rearranging those different components in ways that are interesting. I think that chemistry is fundamentally different in the sense that all the molecules have not been made before. Chemistry is always going to be pushing into new regions of space in a way that is a little bit different, perhaps, than the task. And for that reason, I think that reinforcement learning with human feedback, which is so critical for the success of the GPT models, has to be supplemented in our world by reinforcement learning, not only with human, but also laboratory feedback in our world. So I think that coupling these models to experimental feedback will be something that we always have to do to tamp down on hallucinations and to improve the overall predictions. That's the way I see that, at least for the time being.
Excellent. And Alan, what does the corporate world think of generative AI in the space of medicinal chemistry?
Yeah, so it's quite exciting. If you look at GPT-4 in that paper that was published, it took the AP chemistry test, right? And GPT-3 got like 15% or 15% of the maximal score, and GPT-4 got 80% just out of the box. I don't know what they did to train on chemistry, but that is pretty good. It's pretty much a high schooler in chemistry. And even a lot of us probably taking the AP test now might be getting around 80%. And so it is quite exciting, and we are looking at ways to leverage whatever knowledge it has for giving us fresh ideas and new ideas that are perhaps different from that medchem intuition. And you can call it medchem intuition, you can call it experience, or you can call it bias. And so what we want to do is be able to check those biases with something that's systematic and objective. And I think methods like GPT-4 or other generative approaches allow us to do that.
And Jennifer, from the academic perspective, there's been a lot of discussion about the disruption of large language models and the general academic and publishing model. What are your thoughts about, the applicability, particularly as we think about the language of protein small molecule interactions?
Well, I would have to agree with Tom on the fact that the chemistry space is quite vast, much more than the language space. And I also had the chance to spoke to Eric at the Quantum Nexus at UW recently, where they hope that they could have a the Azure quantum computer, which calculate all kind of small molecules using quantum mechanics, representative, so that they have more data sets. Like we all know that AI is we already depend on the data set. So yeah, with the hope that you can compute faster, accurate, more representative of the potential of the molecule of chemical space that you covered, and you can learn much more. So that is very ambitious, but it's awesome in my perspective, that I believe these, with more data, we have more computational resource, the chemical space that we learned, and the, those kind of interaction that we could soon understand more about the chemistry space. But that also said, in the perspective of understanding the large data set also have to do with the experience and knowledge that one come along with that. Has have a kind of human interface to it to understand, not to bias against certain things. And we are biased creators. So hopefully, if we have more data, we could be able to eliminate those kind of bias along with us.
And final thoughts on this topic from Anthony.
Sorry, mute. I guess it's pretty clear to me that AI does have the power to do a lot of things. I have no doubt, starting with passing an AP chemistry exam, that's great, but it doesn't necessarily get you the whole distance. And even if the next version can then produce configurations or new molecules, and it understands enough chemistry, fantastic again, it's going to be useful. I have no doubt that it's going to corner several niches that right now are the purview of physics-based. But if we want to go after things like new targets and undruggable data sets, or undruggable targets and new data sets, you need that data to teach it. And we don't have that. We don't understand the chemistry well enough to teach the chemistry in a lot of these cases. And so I guess my question is, how far can we expect it to go? It's not going to be able to address problems that we ourselves cannot figure out unless we can build a data set big enough for that. I'd be interested to hear what other people think those limits are.
Okay, Anthony, as you're on deck, there was another couple of questions which were very much on the themes that the panelists have already spoken on. The idea of generative chemistry, and one question was, will models ever be capable of true extrapolation versus interpolation? So this idea of, okay, can generative models forge into new chemical space? Bearing in mind that the rules for chemical space, at least in terms of druggability or drug likeness, are well described. And what is the value of that approach versus mining, in fact, what is already a very large and useful extant molecular environment?
Yeah, I would start with the perspective that I think the real value for us is in exploiting what AI has already laid open for us. Things like AlphaFold have now shown us what the structures are, not perfectly, but shown us enough of the structures of all the proteins that we can now ask, can we go and drug more of them? The question of how much of that AI can address is open and before all of us. I certainly don't know where those limits lie. I think it almost echoes the same thing I've already asked. Where are we going to exploit? What can it do? And can we expect it to do things that we already know that we don't know how to train for? So one day we're going to have to wake up and start dividing this out and say, where do we really think AI will excel? And maybe some of the physics stuff will fall away from that side of it. But overall, I think what Tom said before is right. You need to be playing both pieces together. It's not going to be an either or. Maybe we can train a new AI using the physics models that we're building, and we can then go after things that we previously were considering inaccessible. So again, this is something that we're going to have to figure out as a group. And today we're just on the cusp of understanding the limits of both. AI has really changed a lot in the last 3 years. And even though the physics side has stagnated for the last 20-25, it doesn't mean we can't make more strides on that side as well. So fundamentally, I think we're really at an interesting turning point, and the work that Tom's doing, I think, really demonstrates the value of combining both. They shouldn't be working independently.
Tom, do you want to think about, the ability of models to actually generate not just structures in chemical space, but synthetic routes to those structures? For those of us who spend time mapping existing chemical space, it's pretty clustered around, basically chemistry that has been practiced, over the last 20, 30 years, mostly in industrial settings. There's large swaths of chemical space where we don't have chemistry to access it.
Yeah, I think I want to pick up on the extrapolation versus interpolation point for a second, and I think that's another interesting question. I think that one thing that's really worth noting about extrapolation versus interpolation, I don't think it would be objectionable to say that almost any AI tool is going to be doing interpolation. But what is interesting is that whether you are doing interpolation or extrapolation depends on your representation. So you might be looking at a task with one representation and say, oh, that looks like interpolation, but in a different representation, it looks like extrapolation. A very simple example of that has come in some of our work where we've done, you can do a machine learning model to try and predict properties of molecules that have different elements. And if you train in an atom-based representation, that's obviously extrapolative. If you've never seen a sulfur, how are you going to predict the properties of a sulfur based on only having seen oxygens and nitrogens and carbons? But if you do that in a different representation, if you do that in a molecular orbital representation, suddenly they're all just, sigma bonds and pi bonds and lone pairs, that kind of thing. And it's very trivial. We show lots of examples how you can never have had an element type, even in your training set, and you get fine predictions on those tasks. So I think one of the reasons that these exciting new models are appearing to surprise us is because of the fact that they are starting to learn representations and to see connections that are just higher dimension than what we are comfortable as seeing as interpolation. So therefore they look like extrapolation just by virtue of that learning of the more sophisticated representations. So I think that's interesting. Coming to your other point about, predicting the molecules versus predicting the pathways, it's really kind of two halves of a coin. It's one thing to have a great molecule. It's another to have a great molecule that you can get rapidly into clinic and to scale up and to have nice formulation properties and those different things. So there's a lot of merits to staying close to the boat in terms of chemical transformations. And it's quite striking how much chemical space is actually available and untapped, even close to the boat in that way. So I think that's one general strategy that's worth doing. On the other hand, there's nothing better than these tools that we're talking about today to untap new chemical spaces and to predict not only, reaction energy basins, but also barriers between them that will lead to chemical transformations and predicted path.
Excellent. There were a number of kind of comments that came in ahead of time around cryptic pockets. You demonstrated a nice example of that, as did Alan. Many cryptic pockets are missed by traditional X-ray crystallography, but obviously the explosion of cryo-EM, which now basically allows you to see far greater ensembles of protein structures, may be a clear path to thinking about mining very different protein structural space for cryptic pockets. So maybe Alan or Anthony, do you have any thoughts about using cryo-EM data in a very different kind of way to think about allosteric and cryptic binding pockets?
Go ahead, Alan. I spoke last.
Okay. Yeah, you can go ahead, and I can jump in. Yeah, or I can jump in too. Yeah.
I was going to say you should go first. You probably have.
Okay, sure. Yeah. Yeah. So so cryo-EM is also exciting technology because you can see these ensembles of protein conformations. And you could see the potential of learning off of these ensembles. It gives you a lot more data to build an algorithm. Having said that, Pocket Miner is something that's out now for identifying cryptic pockets. And it's actually pretty decent for cryptic pockets. Obviously, it's learning off of known data, and whether it can extrapolate into newer protein families, et cetera, is to be seen. Yeah, I think that's where, and with AlphaFold and with these large language model type, so instead of doing the alignment that's required in AlphaFold, but using a large language model such as ESMFold or OmegaFold, it's those language LLM-based fold predictors seem to be better at generating diverse conformations. And so that seems promising as well, something we'll track. I think AlphaFold, all of us are probably, finding a lot more time to be creative with things like AlphaFold and GPT. I've been using AlphaFold a lot for homology modeling, and my team has too. And it saves us something that usually takes a week for us to do. I don't know that it's necessarily better than a manual generation of homology models, but it saves us a lot of time. So something that might take us a week would take us a couple hours. With GPT-4, I've been able to code again, because finding those chunks of time, like five or 6 hours, is hard. But with GPT-4 helping you, or a co-pilot helping you, you can now code it something that would normally take 8 hours and probably do it in an hour or 2 hours. So that's exciting to me too.
Jennifer, I see you nodding. Thoughts about cryo-EM and cryptic pockets and unstructured proteins?
Well, cryo-EM is a great tool now, right? Everyone not doing X-ray crystallography anymore to do like cryo-EM. Again, the whole kind of protein ensemble conformations that coming out of these data set from the the database from the AlphaFold or from the cryo-EM structure to study it with, that one can for many different methods that one can use. For instance, hallucinations, or could be able to find out where the kind of the cryptic pockets could stay, or one can use there are many different software, that we can find these cryptic pockets and the replacement of the kind of what we call elastic pocket that only come when the protein is kind of dynamic. It's moving. It's not a static structure. So when you combine these kind of physical based methods like sampling using molecular dynamic simulation or coarse grain simulation, one can find a kind of opening or breathing motion pocket that that could help to find the drug bindings, maybe small molecule that could bind into these kind of cryptic pockets that coming out of the data sets from the cryo-EMs or from AlphaFold structure that you have. So there are other methods available out there that one can use to determine these cryptic pockets, and therefore you can use it for drug targets.
Anthony, last word on cryptic pockets.
Maybe just to reiterate what I said earlier, and I think that in large part, a big failure of the current MD paradigm is that it really isn't great at doing this type of extrapolation, mainly because it doesn't really capture the proper interactions between side chains or between solvent and the side chains. And if we were able to get the physics right, I have a feeling that we'd be able to go after a lot more of these interesting cryptic sites or potentially shallow binding sites and all of that. Again, that's exactly what we're trying to do right now, is to build the tools that allow us to do that. What I find really interesting is that we might be able to use these tools to train AIs to do them. And maybe that does allow us to go after some of the more cryptic stuff in the end. So I haven't worked with cryo-EM, and my experience with AlphaFold is that it still has its pitfalls, but there's no reason why we can't combine these tools to start to mitigate some of the weak points of one with the stronger points of the other. So that's where I'd fall on that.
Maybe can I just add one more thing?
Yes, of course.
Yeah, so if you think about cryo-EM and crystallography, those are both done at cryo conditions, right? And so proteins, of course, in your body are not, hopefully not at cryo conditions. And so we'll have to, it's learning off of cryo conditions, right? Whereas, for instance, physics-based approaches, you can have different temperatures. Having said that, some of these newer, I think the fusion methods have a temperature built in, and there are probably other methods that are somewhat physics-based, have a temperature built in. So maybe there's promise there. Yeah.
Okay, and we're going to finish up with a question that is asked in every one of these webinars. And I'd ask each of you to give a quick answer. And the question is, how much data is required to enable machine learning for trustworthy results? And I'll start with Tom.
Well, I mean, of course, it depends. It depends on the nature of the quantity that you're trying to predict, and it depends on the nature of the quality of the pre-training that you're able to do. Our rule of thumb is that when we're working on a new project, usually with 20 to 40 structures on any given ADME property that we're focusing on, we can have something that will be able to guide synthetic accessibility. But that's because we're riding on the back of billions of physics-based calculations and other pre-training and multitask learning and all sorts of other things that have gotten us kind of to that place. So that's what I would say is the short answer.
Excellent. Anthony.
I'd approach that from a different side. If we can teach GPT-5 how to understand all of chemistry, then we will already know the answer to everything. But I guess I'm not quite that optimistic, but I would say that as long as you have a model that does allow you to test and tells you real world answers, you should be able to use generative AI and then use your physics-based model to filter. Right now, I think we're stuck in the position where you have to generate that molecule and go back to the lab and ask the real world, does it match? And whichever of our models gets us closest to giving us the real world answers will determine where we can stop generating enough data to train those models.
Jennifer.
Well, I'm a big believer of the design circle. That means, the data set you're predicting and you go back to the lab, the combination of a whole kind of integrating on physical-based AI space and experimentations to steer you into the right space, to right the chemical space interactions. So the combination of the whole kind of design circle could data will help us to answer many questions.
And finally, Alan.
Yeah, so I think 3 or 4 years ago, I would have said about 10,000 data points for deep learning approaches to be better than traditional like random forest approaches. But then more recently with approaches, and these approaches are not necessarily new, but active learning, where you can kind of zoom in on a particular area that you care about, it should be a lot less than that. So I'm not giving you a number, but I think with active learning, you can kind of learn about the space around what the area you're interested in. And that might be a good approach. And I think that's an approach many of us are taking.
Excellent. And with that, I'll thank the panel and hand it over to Jesse to wrap up what has been another excellent webinar, and hope that everyone who dialed in enjoyed it and found it as informative as I did. Jesse.
Thank you, Philip. And again, like you, I'd like to thank our panelists, Tom, Anthony, Jennifer, and Alan. Thank you very much. I know from a CQDM perspective, it's always great to have these webinars because I think it really does kind of represent what CQDM does and how we interact with the ecosystem. So we have, we have Canadian kind of biotech and academic research with Jennifer and Anthony, and then we have our pharma partner, Alan, from Merck, who's a critical partner of CQDM and has been for the last 15 years. But also being able to bring kind of an international perspective with a U.S. company from Tom and then, and Philip. So I'm really thankful to all of you to be able to do this, and apologies for the early technical difficulties to everyone, but I think we were able to get through it. And as Philip said, I think it was a great discussion between some real experts in the field. So I appreciate that. So thank you to everybody for attending, and we will hopefully see you later on this year with another episode in this series. Thank you.
Okay, thank you, everybody.
Bye.
Thank you, everyone.
All right. Take care, everyone.